Bismillahirrahmanirrahim. My name is Dr. Mohammad Latif and I am your instructor of the subject Principle of Animal Life 2. Today is lecture number 15 in which we talk about applications of DNA technology. In previous lecture we have learned in detail about recombinant DNA technology, and in this lecture we'll talk about what are the applications of DNA technology. This technology has multidisciplinary applications and potential to deal with important aspects of life. It can improve our health, enhancing food resources and resistance to divergent adverse environmental effects. And in agriculture, it create wonders, and the genetically modified plants have augmented resistance to harmful agents, enhanced product yield, which is very helpful by increasing population of the world, because if we do not have enough crops, we cannot sustain our increasing population. And the other use of this technology is for the purpose of bioremediation and treating serious diseases. What is bioremediation? We talk about in detail about the bioremediation and about treating serious diseases which cannot be treated by ordinary means. We can treat with this technology. And the other use of this recombinant DNA technology is playing a vital role in improving health conditions by developing new vaccines and pharmaceuticals product. So a number of new vaccines are developed, and these vaccines provide immunity against different viral diseases. So we'll talk about in detail about the applications of recombinant DNA technology, and first we'll talk about transgenic bacteria. What is transgenic bacteria? The bacteria with foreign DNA are called transgenic bacteria. Recombinant DNA technology is used to produce transgenic bacteria, and these bacteria have different uses in our society. First of the use of these transgenic bacteria is the synthesis of pharmaceutical products, because a foreign gene is replicated and expressed in these bacteria. Thus, a large amount of protein product is obtained. Many biotechnological products are produced by bacteria. These bacteria are now available in market. Some of these products are insulin, human growth hormone, tissue plasminogen activator, hemophilia factor 8, hepatitis B vaccines. Previously, the insulin are obtained by killing animals and obtained from the pancreas of pigs, goats, cattle, but with this technology we can easily make insulin from bacteria. And the other use of these transgenic bacteria is promoting health in plants. Transgenic bacteria are used to promote health of plants. For example, a bacterium normally forms colonies in the roots of the corn plants. some genes from another bacterium have been inserted into these bacteria. These genes code for an insect toxin, and the toxin protects the roots from insects, so there is not use of chemical pesticides to kill the pest. So the plants itself produce toxins, and these toxins can protect the roots from roots of plants from insects. And the other is biodegradation. I have talked a little about biodegradation. You all must know that bacteria is a decomposer. They decompose the organic matter into simple inorganic material, into simple inorganic matter. And bacteria degrade a particle substance, and the ability of degradation of bacteria can be enhanced by genetic engineering, so we can degrade more material from these bacteria. And the recombinant bacteria or the transgenic bacteria can be used as biofilters. What is biofilter are? Biofilter, you must familiar about them. Pollution, and there are certain type of pollutants which can be released in the environment by different industries. Bacteria have the ability to detoxify the pollutants which are emitted from the industries. So by transforming these abilities through genetic engineering, these bacteria can be helpful to filter our environment from these pollutants. And the other application of recombinant DNA technology is by synthesis of organic compounds. The catalyst act on precursor molecule during synthesis of organic chemicals. Bacteria can be used in place of these catalysts, and these bacteria carry out the synthesis of these compounds. For example, aspartame is dipeptide sweetener. It is known as NUTRASWEET. It is prepared by transgenic bacteria. Basically, it is the natural ability of the bacteria to produce phenylalanine, but this ability can be enhanced by providing bacteria with additional gene, so we can make a sweetener which have 0 calories. And nowadays it is very useful because diabetic patient cannot tolerate normal sugar, so they can use sweetener like NutraSweet to make their diet more sweet. And other use of these transgenic bacteria are in mining industry. You know, the major mining companies are using bacteria to obtain various metals. Genetic engineering enhanced the ability of bacteria to extract copper, uranium, and gold from low-grade sources. Bacteria have natural ability to make bond with these certain metals, and this ability can be enhanced by genetic engineering. and we can obtain certain valuable metals from this technology. And the application of recombinant DNA technology is used in transgenic plants. The plants with foreign DNA are called transgenic plants, and the following uses of transgenic plants. This can be used for pest and herbs resistance. I have little talk about plant resistance for certain insects, but in that case, bacteria are present in root nodules of certain plants, but plants themselves can make resistance for some herbs or pests. Foreign genes are transferred to cotton, corn, and potato, and the cell of these transgenic plants produce an insect toxin, so these plants become resistant to pests. And the other use of this technology in transgenic crops. In 1999, these transgenic crops were planted on more than 70 million acres worldwide. Their total acre is expected to become triple in about 5 years. Means the total production of that crops were higher 3 times by ordinary means. And the other use of these transgenic plants are increasing production of wheat, corn, and rice. And you all know that wheat, corn, and rice are the basic food of ours, and we cannot sustain our life if we have shortage of these plants. And agribusiness companies are also developing transgenic varieties of wheat, rice, and corn to enhance the production of these crops. There is another use of this technology by synthesis of human products. Plants are engineered to produce human hormones, clotting factors, and antibodies in their seeds. For example, an antibody made by corn can deliver radioisotope to tumor cells. So tumor cells can be treated with this antibody. And the other application of this technology is by transgenic animals. As plants are very useful for us, animals are also a food for us, so we can make higher growth rate by these technology. The genes for bovine cattle growth hormone are injected by this technique into many types of animal eggs. Thus, many larger fishes, cows, pigs, rabbits, and sheep are produced by this procedure. And other is the gene farming. The use of transgenic farm animals to produce pharmaceuticals is called gene farming. Many antibiotics hormones are produced by this technology. So a gene of different genes can be inserted in animals, and from animals, from their urine or from their blood, or desired gene can be obtained, and this is a very useful method of producing gene of interest. And the other most important application of recombinant DNA technology is the gene therapy. Gene therapy, the insertion of genetic material into human cells for the treatment of a disorder is called gene therapy. Gene therapy is used to prevent, treat, or cure disease by replacing a defective gene with a normal one using recombinant DNA technology. There are two methods used for gene therapy. One is ex vivo gene therapy and one is an in vivo gene therapy. In ex vivo gene therapy, the gene therapy in which genes are inserted into the cell outside the body is called ex vivo gene therapy. The following diseases are treated by ex vivo gene therapy: hypercholesteremia, or the other diseases can be treated. In vivo gene therapy, the gene therapy in which genes are inserted in cells within the body is called in vivo gene therapy. The diseases treated by these technologies are cystic fibrosis, cancer, coronary artery angioplasty, hemophilia, diabetes, Parkinson's disease, and AIDS. And the other most important application of the DNA recombinant technology is the DNA fingerprinting. You know, the DNA is fixed in every individual. So the genetic fingerprinting or the recombinant DNA technology has also been used in forensic science to identify a criminal suspect using DNA samples obtained from various biological resources at the crime scene, and this test is called genetic fingerprinting. And most of the use of this DNA fingerprinting can be used to found the parentage. The DNA is inherited, thus the fingerprint of offspring resembles the fingerprints of one parent. So it can be used to establish parentage. So this is all about the application of recombinant DNA technology.